So this is regarding a new drug that is mitapivat, which helps to improve anemia in thalassemia. Now the question is asking by which mechanism. So answer to this question is B. That is, it activates pyruvate kinase and thus increases the ATP and decreases hemolysis. But we have to understand the exact reason that what happens, why increasing of the ATP decreases the hemolysis in the thalassemia patient. So first understand this, that why the RBC membrane destruction occurs in cases of the thalassemia, why this happens. So for example, we have alpha chain, we have two alpha chain, two beta chain. Here we are taking into account the example that this patient has alpha thalassemia, so alpha chain are not forming. So now beta chain will get accumulate in this RBC. These are beta chain. So beta chain do two things. First is produces reactive oxygen species, and these reactive oxygen species directly destroy the cell membrane of the RBC. Secondly, these beta thalassemia chain which groups are formed, these groups also destroy the cell membrane of the RBC. So one, by formation of the reactive oxygen species, they destroy the cell membrane of the RBC. Secondly, directly they destroy the cell membrane. Now these are the two ways by which destruction of the membrane occurs. And there is a third way in which decreased ATP production also lead to the cell membrane destruction. Now we have to understand how there is low ATP production leads to the cell membrane dysfunction and cell membrane rupture in case of thalassemia. Secondly, we have to also know that how there is less ATP production in cases of the thalassemia. Now, so first we will see that why there is less ATP production in case of the thalassemia. Now, for example, we have talked about these are the beta thalassemia chain. These beta thalassemia chain are making these reactive oxygen species, and these oxygen species are destroying the glycolytic enzyme. So when glycolytic enzyme are less, they will produce low ATP first. So there is low ATP. Secondly, we have seen that the membrane becomes leaky in the thalassemia. So more calcium and more sodium comes inside, and thus the sodium potassium pump and calcium pump has to work faster to prevent the accumulation of calcium and sodium inside the cell. So what they do, they act at a faster rate and remove this sodium and potassium out to the outside. Otherwise, this sodium and calcium, when they will be present inside, there will also be water associated with it, and there will be increased pressure inside the RBC, which can lead to the rupture of the RBC as it will travel through the blood vessel. Third, now what happens is please remember, there are more reactive oxygen species formation. So the inherent cell tries to fight with these reactive oxygen species. So what does cell do? Cell, instead of moving the glucose to the glycolytic pathway, it moves the glucose to the pentose phosphate pathway. Why? Because pentose pathway produces the NADPH, and these NADPH can destroy the reactive oxygen species. So this is the one advantage of pentose pathway. But the major problem is that in pentose phosphate pathway there is no ATP production. So this further lead to the decreased ATP production. So now with these three reasons, we have understand why there is less ATP in thalassemia. Now we will see what happens when there is low ATP. Now we know that we have a sodium potassium ATPase which acts by removing the 3Na plus to the outside and takes 2K plus inside, and it requires ATP. So when there is less ATP, what will happen? There will be water and sodium accumulation inside the cell, and there is increased cell pressure, and thus these cells will rupture as soon as they will move to the smaller arteries. Secondly, see, there is spectrin actin skeleton. These spectrin actin skeleton in the RBC are required for the cell membrane flexibility. They are required for the cell. This has been also asked that which are the proteins which are required for the cell membrane flexibility. So these are the spectrin actin molecules, and they require ATP for their function. So when there is low ATP, this also destroys the spectrin actin molecules, and when this passes to the